In the global panic of coronavirus pandemic, what do you think about virus? Let's learn a little bit science of virus. What is the virus? Is it a living thing like us or other animals, plants, or bacteria? Textbooks define a virus as a non-living object, but lifelike, as it doesn't have the typical cellular structure. Instead, most of the viruses are extremely small, only possess basic genetic materials like DNA or RNA, and protein codes that protect their genetic materials. As we said, viruses do not have a cellular structure. How can they generate their offspring? The strategy is simple, just hijack the whole cells, like Tabloon parasitic in human intestine or Bernese-Von Gluvonarin tree. Viruses are molecular parasites. The first form of life was estimated to exist on Earth 3.8 billion years ago. Before the appearance of life, it was a long wave to evolve organic molecules, including biomolecules, like nucleic acids and amino acids, the building blocks of life. Nucleic acids of certain sequences could attract specific amino acids to a simple protein, and some protein could in turn protect or encourage their self-replication, or even evolve as tools to rob other nucleic acids. They may not intentionally do that, just because it was a wave to outcompete in a battle of molecular replication. Once an advantage was achieved, such nucleic acids and proteins eliminate other molecules and further upgraded their appearance to the cell structure. Cells works as factory to effectively and massively duplicate the genetic materials by making protein and other tools favorable of DNA replication. This could be considered as the origin of life. However, it was extremely difficult to form such a cellular structure, as thousands of genes effectively expressing proteins had to harmoniously work together. Other than accumulation of subtle evolutions of new functionalities for building a cell factory, like bacteria which could independently complete processes in metabolism, adaptation, and replications. Another wave of evolutions was by hijacking any existing factories. Some nucleic acids freely floating in environments. Once got a chance to enter a cell, they will make use of the cellular factory to replicate themselves. If the ultimate philosophy of perpetual existence is self-renewal, then being a form of life is not the only option. Such nucleic acids, including DNA and RNA, eventually evolve as parasitic molecules in the form of viruses. After a virus enters a cell, to replicate itself, virus shuts down the gene expression of the host cell that severely disrupts the host life. After replication of many copies of the viruses, they lyse the host cells to get released. Living organisms need sophisticated structures to deal with the ever-changing environment and keep alive, to consistently sustain the flow of matters and energy, as well as to evolve complex mechanisms of reproduction, whereas viruses don't need to do this to keep them alive, as they are non-living objects. Everything they do is aim at existing cells around them and figure out ways to enter this cell by passing through the cell membrane. Over 3 billion years, life has evolved in rich diversity. Same for the viruses that diversify in many kinds, targeting at various types of cells. Bacteria do not possess cell nucleus, thus bacteria-eating virus, bacteriophage, only needs to inject its DNA into bacteria, leaving the protein codes outside. Animal cells and plant cells have cell nucleates, thus viral DNA has to overcome multiple barriers to invade the cell until reaching the cell nucleus. Despite cells have evolved dozens of membrane protein to safeguard the engulfing of beneficial macromolecules like protein and reject suspicious-looking proteins from entering, viruses have also evolved the membrane proteins that mimic the beneficial protein to food the recognition protein. SARS-CoV-2, the novel coronavirus causing the COVID-19 pandemic, is an RNA virus. Compared to DNA virus, the single-stranded RNA virus is less stable but more prone to changes by mutations. A portion of the changes will be beneficial to the viruses, making them become more contagious and survive longer, while leading development of diagnostic kits and antibody more difficult. Diagnostic kits works by detecting the viral RNA sequence. Antibody recognize the viral proteins. Once the virus mutates, the changed sequence and subsequently altered protein being translated may void the developed test kits and antibodies. Thus, the whole world is still facing the shortage of accurate test kits and effective vaccines fighting against COVID-19.